All, this is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. Today we have a very interesting research from Harvard Medical School from Boston. My favorite place. I've lived in the Boston area or in Massachusetts for 13 years. My best friend, Dr. Asif Chishti, has done MPHP from Harvard as well. So I love that place. So there is a research from there that is very interesting, and I wanted to share that. Here is the basic gist of the research, the spilling the beans first. The research is the following. Let's say we talk about Pfizer's vaccine or we talk about Moderna's vaccine. What is the protocol today for these vaccines and their effectivity? What we do is this. We inject someone with a vaccine and we let them out. We let them go about their business, their life, and then we wait for a certain number of people to become infected. So some people, for example, 100 people are vaccinated, another 100 are placebo, and now we are observing these 200 people. A few months later, and during that time, we continue to check them for RT-PCR to see if they got the infection. If they did get the infection, then we compare who in the vaccinated group was infected versus who in the, or how many in the placebo group were infected. So this is our way of measuring the efficacy of vaccines. And you can actually see that the problem is we inject someone with the vaccines, then we wait and we wait and we wait for them to get infected or not infected for a bunch of months. So the researchers here, they said, ideally we should be able to, after injecting a vaccine, we should be able to see the antibody levels generated. And then based on that level, we should be able to tell if the vaccine will be useful or not. So you might say that, well, this is logical. Isn't that the right thing to do? And yes, if you look at the vaccines, in the phase 1, or phase 0 in the animals, but in the phase 1, they normally do this, that they administer various doses, and then they look at the blood for the presence of the antibodies. However, we do not know what is the antibody level that is protective. So merely the production of antibodies does not tell us that is it protective or not. That is a problem. So let's say if we together created a vaccine, we are all the scientists together, we worked on it and we created a vaccine. We said, well, our vaccine is going to work. We injected that into a person, and then 2 or 3 days later, 6, 7 days, 10 days later, we looked at the blood of the person and we saw the antibodies present. We said, yes, we have the antibodies, but what we cannot tell is that are these antibodies sufficient to protect from the virus or not? So now how do we tell that? We wait for that person to become challenged by the virus randomly, and then we see if they became infected or not. This is the problem. And the solution is in this study. They are saying for the future vaccines, for example, Moderna, Pfizer, Oxford, Imperial, AstraZeneca, and others, they have done the trials. But for the future vaccines, a faster way will be, if we know what level of antibodies are needed, then you inject the vaccine, you wait for a few days for the B cells to become activated, then you look at the vaccine antibody titers, and then based on that, you can declare vaccine to be useful or not, valuable or not, and based on that, you can approve it to go out in production or not. What does that mean? That means instead of waiting for months and months, for example, these vaccine has been in trials since March, April timeframe till now. So instead of 6, 7, 8 months, you could actually within 2 to 3 weeks declare a vaccine to be effective or not. Safe or not is a different discussion. Effective or not is a different discussion. And then based on that, you could say, all right, this vaccine is approved or not. So that is the basic research. Beautiful research. I loved it. The second part of the research is something that I have been talking about for months now. And somehow the scientific community has been leaving this on a side. And that is, it is not necessary that it is only the humoral arm or the B cells and the antibodies that are protective. The cytotoxic T cells and the innate arm do play a role as well. And we have observed in various studies that if the cytotoxic T cells are playing a role, then the person actually has less symptoms, but they may not have the right antibody levels. So if we have two people, one person has humoral response, antibody response, and the other person has T cell response as well, then the T cell response person may have lesser symptoms, antibody response person may have more symptoms. So they proved that as well, which of course made me happy because I've been talking about it. They proved it that if the antibody is less in amount, however, T cells are functional as well, then the person can still be protected. So these are the two main takeaways. So when we look at the illustrations, what I would request you to take away from there is, number one, these two lessons, and number two, what are the antibody levels? What are these levels which were not known before? Previously, the effectivity was based on statistics, that how many people got infected and how many people not got infected. That was not the level based, that was statistics based approval of a vaccine or effectivity of monoclonal antibody. Today, we will know the levels which are necessary. So let's start with that discussion. So hope everybody is doing good. I am excited about today's discussion. So this is Drbeen.com. And here is the study. The study is published, I think it is a preprint yet, published in Nature. The physicians, Dan H. Baruch, so he is, Center for Virology and Vaccine Research, Beth Israel Dickinson Medical Center, Harvard Medical School, Boston. And he is, I believe, a physician and an immunologist and virologist and so on. And here is his team. This is their study. I have their article open here as well. I had to go through this a few times to actually understand that what levels they have said. And this green paragraph is the soul and the heart of this study. So let me explain it with our illustrations. So here is what is happening. So the question is, what are the protective antibody titers? What are the levels of protective antibodies? And this is useful for vaccines, this is useful for antibodies, and this is useful to test for convalescent plasma as well. So for the first time, this study is presenting those levels. So here is what is happening. So let's say this is us, a human being, and there are various levels of immunoglobulin G that are either given from outside or are produced inside by triggering the immune system by vaccination, or are produced by the disease itself. Here is the SARS-CoV-2. The question is, what are the levels that will protect us from this SARS-CoV-2? That is a question. So let's see the answer. What they did was, the experiment was, they infected a number of rhesus macaque monkeys with the SARS-CoV-2. After the infection, they collected their plasma. And of course, the plasma will contain polyclonal antibodies, correct? Not monoclonal, polyclonal. And why polyclonal? Because the antibodies that are generated are going to be targeted to various parts of SARS-CoV-2. So they took that plasma, then from that plasma, they extracted various immunoglobulin G antibodies and concentrated them. Why did they do that? They wanted to concentrate them so they can inject various levels of antibodies in naive rhesus monkeys and then infect those monkeys and then see what level is protective. So this first part of the experiment was only done to be able to collect the polyclonal antibodies from the convalescent plasma. So why did they not give the convalescent plasma itself? Because the convalescent plasma did not have the amounts that they wanted to give in various doses. So they concentrated them, collected them separately so they can control various doses in various animals. Good. Now, next part of the experiment. What they did was, on day 0, so let's say today, they infected a few rhesus macaque monkeys with SARS-CoV-2. And the way to infect was that they would inject the virus in their nasal cavity and in their mouth and in some cranial cavities and so on. So they injected them. Once they had injected them on day 0, on day 1, of course, by day 1, you are not expecting them to make their own antibodies. Their own antibodies would start forming from 7 to 14 days and beyond, correct? On day 1, so the second day, they started injecting antibodies that they had collected from other rhesus macaques in the first phase of the experiment. And here is what they did. In one group, they injected 250 milligram per kilogram of the antibodies. 250 milligram per kilogram of the body weight of the monkey. So let's say if the monkey was 10 kilogram, I'm just making up a number, then it was 2500 milligram of the antibodies. So they injected 250. Then in another group, they injected 2.5 milligram per kilogram. So 10 times lesser. And then in another group, they injected nothing. Good. So these were the 3 groups. They were all infected. Day 0, infection. Day 1, they got the antibodies. Then they started seeing the response of the SARS-CoV-2 in them to see how much of the SARS-CoV-2 was replicating. So let's look at the result. Those monkeys that had not received the antibody at all and were infected, they started developing or started having the virus in their bronchoalveolar lavage. What they do is that they, and it is done on humans as well, you send fluid or water in the lungs, and then you extract that water, and then you test that water for various components that you're interested, and it is a bronchial fluid wash. So it brings the cells and the chemicals from inside the lungs, and it gives you a picture of what is happening in the lung. So bronchoalveolar lavage in the macaques that were not given the antibodies, 7 to 10 days they had the virus, and 10 to 14 days they had the virus in their nasal area. Then those that were 2.5 milligram per kilogram that were given IgGs, imagine these IgGs were produced by their own body, but they were experimenting by giving the dose from outside in a controlled manner. So in these macaques, they saw that for 3 to 10 days, bronchoalveolar lavage showed the virus. So virus did produce in them, but the production was slightly different. And then in the nasal, from 7 to 10 days instead of 10 to 14 days. So there was some protection, not significant protection. And then those macaques that had 250 milligram per kilogram body weight of immunoglobulin, they had no SARS-CoV-2 replicated at all in the lungs or in the nose. So they were protected. So what were the levels then? What were the antibody? So we give 250 milligram per kilogram from outside. Now, what does that mean? Once that antibody is dissolved in the body tissues and in the fluids, what is the level that is established inside the fluid, which we can take away as a learning to say, when you give a vaccine, the vaccine should be able to produce antibodies with the following levels. So here are these levels. I think this is the soul of this whole research. This is a breakthrough for all of us at this time to understand what is the level. And I can tell you from my own experience, many of my students, medical doctors, students who are medical doctors, they ask me very often, what is the level of antibody in the body that we can, when we are reported that, we say it is good. And you know that when you do the antibody test, at some point they say reactive. They don't say neutralizing. They don't say protective. They simply say reactive. Because we actually do not know what are the levels, what are the right levels where we are protected. So here, for the first time, if it is a neutralizing antibody, so when the antibodies are produced against the SARS-CoV-2, the antibodies are of various types. And we have done this discussion in the past. For example, if I am SARS-CoV-2 and my hands are my spike proteins, if there is an antibody that is against the spike protein, then that is a neutralizing antibody because it has disabled my hands. I cannot function anymore. But if there is an antibody that is sitting on my head and doing nothing, then that is not a neutralizing antibody, although it is an antibody, but it is not doing anything to me. So neutralizing antibodies, 50 threshold or titer level 50 of the neutralizing antibody is protective. Receptor binding domain, ELISA threshold of 100 is protective. S or spike protein, ELISA threshold of 400 is protective. So that is what, let me show you here as well. So if you see here, we employed logistic regression models to determine the antibody titer thresholds for protection against SARS-CoV-2 in this experiment. These regression models indicated that pseudovirus N neutralizing antibody titer of approximately 50, RBD ELISA titers of approximately 100, and S or spike protein ELISA titer of approximately 400 were required for protection. Beautiful, beautiful response here. So just very quickly now, if I go here, what are these threshold mean? What do they mean? So if this is the virus, this is the spike proteins. We know if I make one spike protein rather large in its structure, we know that the spike protein in turn has receptor binding domains on it. These are the areas that will bind with ACE2 enzyme, correct? So this is the ACE2 enzyme on our cell. So what happens is this, if there is an antibody against the spike protein, we will call it S-ELISA threshold. If there is an antibody against the receptor binding domain, that would be RBD ELISA. And if there are antibodies that are just connecting in general with the virus and neutralizing it and not letting it function, may they be for the spike protein or RBD or other parts of the virus, these would be called neutralizing antibodies. So they have titers for all of them. This is the first time. I love it. If I could go to this researcher and that group and shake their hands, I would do that, because this is an important breakthrough, not only for SARS-CoV-2, but in the future for making more vaccines or monoclonal antibodies, or to figure out what is the right level for protection from some virus of the antibodies. They can employ this method as well. And it's a very simple method. And finally, the last part of the discussion today, role of the cellular immunity. And I am so pleased that I've been talking about this for months now. Remember this diagram that I've been making, that there is innate arm that functions, and then there is humoral arm that functions, and then there is cytotoxic arm. This is what they're talking about. So what are they doing is this. They're saying, when we give immunoglobulins, And even if the levels are low, however, if the CD8 T cells are competent, then the macaque had the protection. This is where some people, when they say, my antibody levels were not great, but I still was with someone. Even yesterday in the open forum, somebody said that my wife and myself and my child, we lived together. And then I think she got they all became COVID positive, and they all had the antibodies done. And she had positive antibody titers and they did not. And what is the reason for that? And so the reason for that is that the CD8 T cells were functioning. In the child, it is possible that the innate arm was taking over. In the adult, it is possible that not only the antibodies, but the CD8 cells were working as well. So here they saw that the macaque that was CD8, that had competent CD8 cells, was able to work and protect itself even when the antibody levels were low. That means even when B cells were not functioning that much. And we have actually seen it that if B cells actually function less and CD8 cells function more, the person handles the SARS-CoV-2 with less symptoms. So that they proved that. And what they had done was they also took some macaques and they had deleted their CD8 cells. So they sent in antibodies that would go and kill the CD8 cells from them. So they made them immunocompromised by removing their CD8 cells. And then they gave them the antibodies and made them infected as well. And these macaques were not able to handle the virus that well as the one with the competent CD8 cells. That is a huge deal. So this is the important part. So I would just now wind up by creating a diagram here. So let's say this is the innate arm. Innate arm. Somebody is saying, is that a helpful video? I have no idea. Maybe it is helpful. Okay, so let's say this is the innate arm. And this innate arm, let's say this cell here is a macrophage, or it is a dendritic cell, or it is a neutrophil, something, or natural killer cell. Then we know that let's say this is a dendritic cell, it connects with a naive T cell, correct? We have been doing this for a long time, naive T cell. Naive T cell is like, I do not know anything in my life. I do not know my purpose. Then we go to T helper 2 pathway that goes to B cell becoming a plasma cell. Correct? And then that plasma cell releasing antibodies. And then it is also possible that this pathway, this naive cell, so this was interleukin 4 or absence of interleukin 12. This is interleukin 12, presence of interleukin 12 causes the naive T cell to become T helper 1 cell, correct? Which T helper 1 cell in turn releases interleukin 2 and causes cytotoxic T cell, the angry T cell, to become activated. This cytotoxic T cell is also called CD8 plus cell, right? And this is the one that kills the other cells. What the researchers are saying is the following. If the B cells are not functioning as greatly, but they are functioning, they are making some antibodies. However, the CD8 cells are also functioning, then the person is protected. So it is not necessary that just this part works only. They both can work together as well. And we know that in children, this part works more. In some adults, this part works more. And in others, this part works more. And they all have varying things in their disease progression. Children, no symptoms. Adults with this pathway, fewer symptoms, even asymptomatic. And adults with this pathway, antibody production, cytokine storm can occur as well. So this is the discussion for today. I hope you are you enjoyed it. I'm going to bring it back to this because this is the soul of today's discussion, and I would stop here. So let's look at a couple of comments, and then tomorrow is my off. And yes, I wanted to thank everyone. Nina's fundraiser has been completed. Janet, Margaret McInnes, Dr. Tipu, they have all donated more, 500 each, or Margaret 600, and then lots of other folks have donated so much that we have actually exceeded the fundraiser milestone. So thank you very much. Please do not donate more for Nina because her milestone is done. We'll continue with the book as well. So thank you very much. You are really cool beans. You're helping. I have called Nina a couple of times. She did not respond today. Maybe she was busy. I would let her know and transfer the funds to her for her insurance. So thank you very much for helping me protect someone, giving them hope. This is what is my belief here as well, giving you hope and giving you some idea for how to protect. And you helped me do the same thing for one more person. So thank you. So there are some questions. JTJ says, I'm wondering what to do for someone who has recovered from COVID-19 but is having memory loss, brain fog, and fatigue. JTJ, I did a video about it. Myalgias, brain fog, fatigue with this. Please watch that video and then maybe tweet at me and we can discuss further. So Brown Hilda Santos says, can you explain the difference in response between the 2.5 and no IgG again? Thank you for the excellent talk. In case of no IgG, of course, no protection. In case of 2.5, the protection was insignificant. What they wanted to prove with that was if the antibody levels were low, they were as bad at the response of the body to protect from SARS-CoV-2 was almost as bad as if there was no antibody. That is what they were trying to prove. So if I share that for a second, what they said was, if you see here, this is 0 milligram, correct? So let me take the comment out 1 second. So if you see here, 0 milligram, the macaque had the virus replicating for 7 to 10 days in the lungs and 10 to 14 days in the nasal swab. And 2.5 milligram offered so low protection that 3 to 10 days in the lungs and 7 to 10 days in the nasal swab, meaning the virus continued to replicate in them. So this was not a good dose. And then those that were at this dose, they had no virus at all. So then they proved that this dose, whatever antibody titers it created in the body, were the right titers. And that is these titers. They do say in their studies that it is possible to further refine this and then start reducing it to, let's say, 200 milligram and then see what happens, or 300 milligram and then see what happens, or 100 milligram. So this can be further refined, but they have figured out that 250 is the right dose, at least in this study. So I hope that answers your question. Going back to the questions here. So share, at least for the time being, the vaccines that are given out, they have been tried on 18 plus, so children are not part of this. Yes, Barbara, the link for the book, why not I do this? I would tweet the link. I'm sure I can very quickly get it from here as well. Let me get it, and I would put that in the comments here. So for the book, here is the link. Just 1 second, please. Barbara, I'm posting the link here in the comments, and that is a link for the book's handbook. And thank you very much for supporting there as well. What is... So Barbara, I just commented it here. BLSR says, Dr. Marek has a note in his EVMS doc that a study shows ivermectin is good for long haulers. Yes, it is good for long haulers. But I have seen steroids to be even better because ivermectin does the modulation as well of the immune system. Steroids do that too, and steroids are much more powerful. How important are antibodies if they don't even show up till 3 weeks post-infection on tests? I think we overestimate antibody and underestimate T helper 1 and innate arm. This is the innate arm and cytotoxic T cells do have their own benefit. However, antibodies always start anywhere from 7 to 10 days to 21 days or so. That is their window of starting to produce the results. Yes, you are correct. We have not paid attention to the cytotoxic T cells and the innate arms behavior. And you are very correct, John. When I started for the book and for Nina, in the beginning, there was like $50, $60. And I was feeling that maybe it will not happen. And then within a day or two, Nina's milestone was met just like that, 10, 15, 5, 50. And so it happened. And for the book as well, it is happening. So you're correct. Is there lab equipment that you put samples in and they give milligram per kilogram out? So what they do is they take the concentrated antibodies and then they take the fluid in which they're going to put it and then they add them. They can also take a sample of the fluid and then put it under the microscope and color it and then see what is the density in there. And from there, they can estimate the dosage. So yes, there are many lab methods and equipments that help with that. You are very welcome, Professor Mulazim Hussain Bukhari. You are my teacher as well from King Edward. So thank you very much for joining and listening. I am actually your student. So Wayne says, Dr. Bean, can ivermectin be taken to combat other viruses? Yes. So if you see the ivermectin's behavior of disrupting the viral cargo to nucleus can be used for other viruses that do that. Ivermectin's behavior for inflammatory modulation or the immune modulation can be for other systems as well. So it is a broad spectrum antiviral. 008 says, I will love this video after I watch it 3 to 4 times more times. I'm trying. Thank you very much. France says, yes, that's great. So happy for Nina. Absolutely. Thank you to all the cool beans. Cool. So this is what we have. There is a question, one more question here. Sepil Steve says, can effective levels of antibodies in vitro be relative to how they will work in vivo? I believe you're saying. So very good question. And I should have addressed that. The study says that these are the levels in macaques. We still need to kind of figure it out in the humans. So they say there is further, they've given a method, they've given a way to do this, and we can use that as the initial samples, and then we can refine it. So yes, your question is correct that the in vitro, can it go to the in vivo? Although this is not really in vitro, this is still an animal study, which is very closely related to humans. So this study is much more related to human as well. In vitro can be taken to human as well, but that would need to do the phase trials. All trials start with in vitro, then phase 1, 2, and 3, and then production is phase 4. So Kabaili CD Ike says, can we utilize this monoclonal IGs as a drug? Yes, as a therapy, correct. And this study over here and these numbers that I'm showing are actually also a guideline for monoclonal antibodies as well, that what should be the monoclonal therapies amounts. Gaurav says, if after 5 months of having COVID-19 antibodies are gone, what does it mean? Will this mean vaccine will provide lasting immunity? So we have talked about it many times. Antibodies decline naturally because the cells are sleeping. And so they're not functioning. They're just releasing small amounts of antibodies. Plus, in the case of SARS-CoV-2, it is possible that innate arm was strong or the cytotoxic arm was strong. So reduced antibody levels is not a big trouble. If we get reinfected, in theory, those B cells will wake up and they'll start making antibodies, but within 24 hours instead of waiting for 10 to 20 days. So that is the benefit of the memory cells. So Jim is saying, where's Luffy? So that is surprising. I think Luffy went up with my wife. She was doing some painting and he went with her. Luffy is, he would go with whoever talks with him. Luffy must be tweeting somewhere, correct? So guys, please do me a favor. Please like, subscribe, and share if you like the work. And if you wanted to support my work, there's a link in the description for donations. And if you would like to have these things in a book as well, I have a GoFundMe as well for the book. So thank you very much. Tomorrow is my off. I would see you all on Monday. Please stay safe and healthy. Please keep exercising the social distancing and masks. Vaccines are coming. Whoever out of you are convinced for the vaccines, I am, at least that would improve the overall situation outside. So hang in there. Don't become frustrated yet. We are almost there. So see you on Monday. Bye-bye.